Now, let's see the various options available in multiSLIDE for ordering samples. To demonstrate the multi-level phenotypic sorting in multiSLIDE, for this video, we will use the time course data studying the mRNA and protein level response to misfolding stress. This data is also available as a demo in multiSLIDE's homepage. The ordering and filtering panel has three sets of controls. The first control is for sorting samples. The drop-down list under Order Samples By presents two options: Sort by phenotype and Sort based on hierarchical clustering. Notice that by default, the samples are ordered by phenotypes. In this case, since we selected replicate as the first phenotype, they are ordered by replicates and then by time points. Notice here, both are sorted in ascending order. The Legends panel describes the phenotype colors. Notice that time point, which is a continuous variable, uses a gradient color scheme, whereas replicates, which is a binary variable, uses the orange and blue color scheme. To change this default ordering, first select the Sort by Phenotype option from the drop-down menu. Then, to specify which phenotypes are used to sort the samples, click the Settings icon next to the drop-down list. You can specify up to 5 phenotypes in the panel here for multi-level sorting. Let's now change the default sample ordering to first sort by replicates in ascending order and then by time points in descending order. We can now see that the new ordering with the time points in descending order has been applied. We can also perform hierarchical clustering on the samples to get an ordering based on the expression values. To do this, we will select the hierarchical clustering option from the drop-down menu. This already applies a hierarchical clustering with the default parameters. We can see that hierarchical clustering reveals structure in the data. To change the clustering parameters, click the Settings icon next to the drop-down list and select the Specify option. The first parameter is the data set. In multiSLIDE, we choose one omics level on which to perform the clustering and get an ordering, and then apply the same ordering across all omics level. For instance, now we will apply hierarchical clustering on the protein data. For now, we will leave the other parameters with the default values and click the Save Changes button. Notice that the ordering resulting from this change is applied to both the mRNA and protein data. Going back to the settings, we see that there are other parameters of hierarchical clustering, such as the linkage function, the distance function, and the leaf ordering that can be customized as well. The time point phenotype in this particular data set already has an inherent order. Therefore, even though reordering the samples based on hierarchical clustering brings samples with similar expression profiles closer, it may not result in the most optimal visualization. We will therefore now go back to using the phenotype-based sorting. However, for datasets that have many samples or for datasets where samples do not have such an inherent ordering, clustering is often the key to revealing structure in the data. The second control in the ordering and filtering panel is for ordering genes. The drop-down list below, Order Genes By, presents three options: order by gene groups, order based on hierarchical clustering, and order by significance level. The first option, groups genes based on their membership to pathways or GO-terms. This is the default option, and we can see this in effect in the gene group tags. Genes belonging to the pathway response to endoplasmic reticulum stress are grouped at the top, followed by genes belonging to the pathway protein processing in endoplasmic reticulum. Now we will change the gene ordering to hierarchical clustering. We will select the hierarchical clustering option from the drop-down menu. This will apply the clustering with default parameters. To change these parameters, click the settings icon. Here too, the same options are available as in the case of sample ordering. Next, we will see the gene ordering and filtering options related to differential expression analysis.